Mr. Chairman, ladies and gentlemen, thank you. This is a trial which began in the mid-90s, so this is data that's been a while coming, and I'm very pleased to be able to share it with you. Aspirin has been associated with cancer chemoprevention since the late 1980s, and there have been a series of studies looking at people taking aspirin or aspirin-like drugs for things like arthritis, which is associated with a significantly lower incidence of colon cancer in these populations. Whether we look at polyp adenoma data, or whether we look at cancer incidence, or whether we look at cancer-associated mortality, people who take a lot of non-steroidal anti-inflammatories seem to die less often of colon cancer. And so we decided that we would set up a randomized controlled trial to test this hypothesis in a very special group of patients. 16 years ago, almost to the day, I was contacted by Richard Kolodner's group, who are yeast geneticists working in Boston, because they had a hunch that the patients that we were investigating with hereditary colon cancer might have a problem in a class of genes which they were studying in yeast, and so it proved to be. So this family was actually the first in the world in which a mutation was identified in a gene called MSH2, which is part of a class of genes which are effectively the body's spell checker. These genes check as your DNA is replicated to make sure that the new copy is an exact replica of the old. And if the genes don't work properly, then you accumulate mutations at a very much higher rate. And that manifests as a very rapid progression to cancer, particularly colon cancer and endometrial cancer, or womb cancer, as has happened in Ted's sister. And you can see here that Ted in fact had colorectal cancer 3 times, skin cancer 9 times in his lifetime. And the young lady at the bottom of this graph here was actually 18 years old when she developed her first colon cancer. So we thought this was an interesting group of people in which to try out chemoprevention, because they were highly motivated, already under surveillance, and were progressing rapidly to a cancer which was nevertheless a mirror of what we see in the general population. And even in its own right, it's a very important condition. Around about 3 to 5% of tumours like colon cancer and endometrial cancer are attributable to this particular class of gene defects. So in England, or in the UK and Germany combined, my colleague Gabriele Merslein and my co-author, who's the German lead, we were calculating this morning about 8,000 people a year getting cancer as a result of this condition in our 2 countries. Our study involved over 1,000 people. Our power analysis said we needed 1,000. We went to 43 centres in 16 countries to collect them. And what made this a special study and found its way into the New England Journal was that we were able to base it almost entirely on people who had undergone genetic testing. So these were people who carried a mutation in the gene, even though they did not yet have the disease. A proportion of them had developed a cancer and were therefore recruited on clinical grounds. And we reported in the New England Journal in December of last year that the trial itself was negative, despite the fact that we'd given all of these people, beautifully randomized into the 4 categories, these 2 interventions, 600 milligrams of aspirin and or 30 grams of Novolose, which is a digestion-resistant form of starch. Neither of these interventions appear to have had any impact whatsoever on the progression to cancer in these patients. But the observational studies argued that the progression to cancer was a very slow one, and so we'd previously consented our patients to allow us to continue to follow them after the study. Unfortunately, the funding agencies didn't think it was worth it because there was no effect in the trial, so why bother? Well, we did it anyway. And what we found was that after the trial, people stopped getting cancer. So in this graph here, we've so far managed to track down about 80% of the participants who went right through the trial, 711 of the total recruits. And as you can see in this graph, 52 people have now gone on to develop colon cancer, which is interesting because these people are having colonoscopy every 1 or 2 years. So we didn't expect this many colon cancers. And as you can see, the number of people who were taking aspirin was only 17 compared to 35 who were not taking aspirin. Now, at face value, that's only a trend. The p-value is 0.08. But in fact, if you ignore the beginning of the curve, bearing in mind that these people have probably almost certainly already had their cancer when they started the trial, then in fact there is a significant impact beyond 2 years. And this doesn't take account of the fact that 6 of these people developed multiple cancers and 5 of those were in the placebo group. What was even more striking was that we also looked at the impact on endometrial and other cancers, and we found that independently it was the people with endometrial cancer were significantly fewer if they'd been taking aspirin. So when you combine all of the data, which was our original analysis, was to look at Lynch syndrome cancers, you can see from this graph that despite the fact we stopped giving them aspirin at the latest 4 years from the start, and the mean duration was only 29 months, remember, the impact was felt for 5 more years. So these people who were taking 2 aspirins a day for 3 years had a significantly reduced incidence of cancer for the next 5 years. So in conclusion, Mr. Chairman, 600 milligrams of aspirin reduces by half the colon and endometrial cancer risk in people with a mismatch repair gene defect. It takes about 3 years to start, but even though you stop taking the aspirin, the effect persists for 5 more years. So our assumptions on why non-steroidal anti-inflammatories prevent cancer need to be re-examined, because most of those are based on the idea of preventing the inflammatory enzymes COX-2 and so on. And clearly it's having some effect on the precancerous state that we still don't fully understand. My hypothesis is that as in plants where salicylate induce programmed cell death as a defense against infection, it may be that a salicylate-rich environment is inducing programmed cell death among genetically abnormal cells in the stem cell compartment that have yet to begin to become a cancer. And only when adenomas expose them does that benefit start to demonstrate itself. Thank you for your attention.